Hello, I'm Dr. Mirko Pegoraro, and this is another lesson of your developmental biology module. Today we are going to talk about paraxial mesoderm. So what are the objectives for today? The objective is for you to be able to discuss and explain how the paraxial mesoderm produce somites, and later on how somites produce their derivatives. Just to remind you, the lesson that you should have had before this one is about neural cell development with Dr. O'Reilly. And in the next lesson, we will talk about intermediate and lateral plate mesoderm as well as the endoderm. This lesson is linked with the chicken and zebrafish practical laboratories. Okay, last time we had a lesson, we talked about the embryo development. I told you about the anterior-posterior axis formation in very early embryo development. Then we talked about the derivatives of the ectoderm. So we talked about neurulation, so formation of the neural tube, and we talked a bit about the epidermis, which is the external part of the body. Now, today we are going to talk about mesoderm, and in particular paraxial mesoderm. Okay, so in this picture, these two pictures actually, you see a representation of a chicken embryo. So this is a cross section of a chicken embryo. All right, so you see here, this is the middle part of the chicken embryo, and these are on the side, the two lateral parts of the chicken embryo. In the middle here, you recognize the neural plate, and below the neural plate, the notochord. And you see externally the epidermis. So epidermis and neural plate, remember, are derivatives of the ectoderm. What is the ectoderm? If you remember, the ectoderm is the external germ layer that the embryo form. In the middle, we have the mesoderm. And here is the mesoderm in this chicken embryo, you can see in the middle. And below that, you have the endodermal layer. Now, the mesodermal specification, or if you want the different derivatives of the mesoderm, are formed at the same time of neurulation. So while the ectoderm is busy doing neurulation, the mesoderm is busy in producing the different part of the mesoderm. In this lesson, we are going to use examples to understand how the mesoderms produce the different derivatives. So the different examples come from mammals, zebrafish, chicken, and xenopods. But all of this example works perfectly to describe mesodermal formation in mammals. So the mesoderm is divided in three main groups, right? It's divided in three parts. The part that are near the neural plate, near the neural tube and the notochord, is called the paraxial mesoderm. So the paraxial mesoderm is the part of the mesoderm that is near the neural plate or the neural tube. The part of the mesoderm that is more far away laterally to the neural tube is called the lateral plate mesoderm or lateral mesoderm. In between the two, you have the intermediate mesoderm. So why do we need to call them three different names if all the cells are mesoderm anyway? Well, it is because this different group of mesodermal cells are going to produce different part of the adult body in the embryo, during development of the embryo. In particular, the paraxial mesoderm is going to form the somites. And here you see these tubes, almost tubes, this structure that form the somites near the neural tube. The intermediate mesoderm form all the structure here that you can see in between the paraxial mesoderm somites and the lateral plate mesoderm, while the lateral plate mesoderm form again different structures, in particular the column, and we are going to talk about the column and the structure that derive from the lateral plate mesoderm in the next lesson. So today we concentrate on the derivatives of the paraxial mesoderm. So we are talking about the formation of somites. Somites are formed from what is called the presomatic mesoderm, or PSM. The presomatic mesoderm is indeed the paraxial mesoderm that is going to produce the somites. How do the mesodermal cells know whether they need to become paraxial mesoderm, intermediate mesoderm, or lateral plate mesoderm? How do they know? Well, they know because of two opposite gradients. The first gradient is a gradient of BMP signaling, BMP4 but other BMP as well. So BMP signaling that are highly expressed in the most lateral mesoderm, so in the most lateral place, we have highly expressed level of BMP, while near the neural tube, the level of BMP is very low. We also have an opposite gradient of nogging. We already know, for example, that the notochord express nogging. So we have high level of expression of nogging near the notochord and low level of expression of nogging far away from the notochord. Moreover, the presomatic mesoderm and the paraxial mesoderm indeed express high level of nogging too. So what's happened is that if the mesodermal cells are experiencing high level of nogging and low level of BMP, they are going to produce, they are going to become paraxial mesoderm and then produce somites. If they have high level of BMP but low level of nogging, then they will become a lateral plate mesoderm. And if they have intermediate level of BMP and nogging, they become intermediate mesoderm. Okay, so these are these two expression profile of BMP and nogging give coordinates, if you want, to the cell to know what they have to become. Now, okay, you heard me already saying a few times the word somites. But what are somites? Well, somites are a block of epithelial cells that are formed from the paraxial mesoderm near the neural tube. Indeed, while the neural tube is forming from the neural plate, the somites are also forming near them. Another important mesodermal structure that we need to mention here is the chordal mesoderm. What is the chordal mesoderm? It's the mesoderm that produces the notochord. Okay, so it's very easy to remember this name. It's the mesoderm that produces the notochord. That's why it's called the chordal mesoderm. The notochord, if you remember, is important for the induction and formation of the neural tube. But at one point, most of the notochord get reabsorbed by apoptosis, but not all of it. Some part of the notochord remain and going to form the nucleus pulpus. What is the nucleus pulpus? Is the part of the body that is going to form the intervertebral discs. We are going to see how the nucleus pulpus is important for the formation of the column and the vertebra in a minute. But basically, part of the chordomesoderm, the notochord, remain and form the nucleus pulpus that end up developing into the intervertebral discs in our vertebral column. Okay, let's have an overview about the derivatives of the mesoderm, or the different parts of the mesoderm. So, today we focus on the paraxial mesoderm. The paraxial mesoderm is going to form the somites, and we are going to talk about the somites in details in a while, because the somites also produce different derivatives. So the cells of the somite are responsible for the formation of different things, and we are going to explore that. The intermediate mesoderm is going to produce the urogenital system, including kidney and gonad. The lateral plate mesoderm, on the other hand, is going to form the heart, the blood vessel, and the body cavities. Cool. Now, finally, we can start talking more specifically about the somite. So you can see here, this is the neural tube, this is the notochord. Near the neural tube, you have the paraxial mesoderm that is going to form the somite. Okay, so the paraxial mesoderm is responsible for the formation of the muscles and the connective tissue of your back. Okay, and this includes the dermis, and if you remember, the dermis is that layer just below the epidermis. Then, of course, the back muscles, but also the vertebrae and the ribs. Okay, if you think about it, the ribs are connected to your vertebrae, so they start from your back, so the dorsal part of your body, and they move then anteriorly to fuse anteriorly with the sternum, not anteriorly, sorry, ventrally with the sternum. The somite is divided in, the cell of the somite can be divided in two groups of cells. One is called the dermomyotome. The dermomyotome, you can see this in this position, in this sketch. This sketch is quite useful. You have here the neural tube cells and the notochord cells. Then you have all the somite cells. So this triangular shape corresponds to all the somite cells. So the most external and dorsal cells are going to form the dermomyotome. What is the dermomyotome? The dermomyotome are the cells that are responsible for the formation of the myotome, which are muscles, and the dermis. Again, the dermis is that layer that is just below the epidermis. All right? Then the rest of the cells, this one here that are color-coded in blue, red, green, and yellow, are going to form what is called the sclerotome. Okay? The sclerotome is important and is responsible for the formation of the vertebra. So this is a sketch, if you want, a representation of a vertebra, one of your vertebra, and not only the vertebra but also the ribs. Okay, so in the sclerotome we recognize different kind of cells. This yellow cell here in the middle are called the arthrotrome. And these arthrotrome cells are responsible for the formation of the vertebral joint. So this joint here, so this is the joint of the vertebra called PE and TPE, right? And the and the proximal part of the ribs as well. So the arthrotrome cells are responsible for the formation of these two groups, these two regions of the vertebra. And remember, the vertebra is made of bones, so the arthrotrome is responsible for the formation of bones. Basically, actually, all the sclerotome is responsible for the formation of bones. And here in this vertebra, you recognize this red part, which is the body of the vertebra, the joint, so this yellow PE and TP, the proximal part of the ribs, and here in green you have the distal part of the ribs. The distal part of the ribs begin from the back, so from your dorsal part of the body, and fuse frontally, fuse here ventrally with the sternum. Indeed, the distal part of the ribs is formed by this group of cells, color-coded in green, which are called the ventrolateral sclerotome. It's easy to remember why they are called ventrolateral, because they are in a lateral position and they are in a ventral position. Okay, so ventrolateral sclerotome, which is responsible for the formation of the distal part of the ribs. These blue cells here, color-coded blue, are responsible for the formation of the arch and the spine of the vertebra. They are called the dorsomedial sclerotome. It's easy to remember because they are dorsal cell and in a position in the middle, so medial position. So if you think about it, this is the center of the embryo. Here you have the neural tube, so these are in a medial position. These red cells here, color-coded red, they are called ventromedial sclerotome. These cells are responsible for the formation of the body of the vertebra itself, and they are near the notochord. Finally, the notochord itself is going to form the nucleus pulpus, and the nucleus pulpus will develop later on in the intervertebral discs. Now, interestingly, part of the arthrotrome is specialized for the formation of tendons. And these cells here then are called syndetome. And if you think about the position of these cells, make total sense. because the arthrotome and the rest, all the arthrotome and the rest of the sclerotome are going to form bones. The dermomyotome, at least part of the dermomyotome, are going to form muscle. So now you need to attach the muscle to the bones. So you need tendons to do that. So the cell in between the cell that form the muscle and the bones need to form tendons. So the syndetome is in between the cell that form the muscle of the dermomyotome and the cells that form the bones of the sclerotome. Okay. Now, how we define the somites in the anterior-posterior along the anterior-posterior axis? So somites are always formed bilaterally at both sides of the neural tube at the same time. You can see here in this sketch, if you want, in this representation, this represents a chicken embryo. So from the top, so here you have the neural plate, the neural plate forming the neural tube. Laterally to the neural plate forming the neural tube, you have the somites. This is the anterior, and here you have the posterior, which is also called the tail bud. For this, when we study the somite, we call this the tail bud. What we have here is that the cells need to decide, the cell that enter the primitive strip here, they need to decide whether to become neural tube or to become mesoderm. Now, the decision is made via the opposite gradient of retinoic acid and FGF. Okay, we already talked about it during the anterior-posterior axis formation lesson. We already know that retinoic acid form a gradient, an anterior-posterior gradient, while the FGF form a posterior to anterior gradient. So there are two opposite gradients of retinoic acid and FGF. Moreover, FGF and Wnt signaling are forming a positive feedback loop with each other. So they induce the expression of each other. Wnt signaling also form a gradient from the posterior to the anterior. We remember that, and retinoic acid actually are counteracting those signaling. So the retinoic acid is formed mostly from the neural plate. So the neural plate cells express retinoic acid, but also somites cells are expressing retinoic acid. This is quite important because the mesodermal cells in the presomatic mesoderm do not really express retinoic acid, but when they become somites, those cells start to express retinoic acid too. So retinoic acid in general induce the formation of neural cells by repression of FGF8 signaling. However, the FGF8 from the posterior will repress retinoic acid, specifically inducing the expression of, CYP26. CYP26 is a repressor of retinoic acid. So therefore, in the posterior part of the embryo, FGF8 induce the formation of mesodermal cells. So a balance between retinoic acid from the anterior and FGF signaling from the posterior will decide whether a cell will become neural tube or will become mesoderm. It is worthy to remember here also another important feature of gene expression along the anterior-posterior axis, which is what is called the OX clock, or if you want, the temporal collinearity of the OX genes. If you remember, the OX genes are organized in the chromosomes in a way that the more anterior genes in the chromosomes are expressed more anteriorly in the anterior-posterior axis, along the anterior-posterior axis in the embryo, but are also expressed early during the development. So this differential expression in between OX genes in terms of their position in the chromosomes that correspond to the expression profile both physically and along the anterior-posterior axis, but also temporally with the anterior genes expressed earlier than the posterior genes, is called temporal collinearity. Again, genes that are physically in the chromosomes more anteriorly, OX genes I'm talking about, are expressed more anteriorly in the anterior-posterior axis, but also earlier during the development. So if you want the opposite of that, the most posterior OX genes are expressed more posteriorly in the anterior-posterior axis, and they're expressed later during development. Okay, what does it mean? This also implies that the cells that enter the primitive streak later, okay, and more posteriorly, will express more caudal OX genes than the cells that enter the primitive streaks more anteriorly and earlier during development. Okay. So this is a beautiful pictures about the formation, which detail the formation of somites in a chicken embryo. You have here the neural tube, and yes, you have a number of somites that are formed bilaterally, and you can see here a somite that is in the process of forming. This structure here, this tissue here, is called presomatic mesoderm, because it is the mesoderm that is going to form the somites. Now, how the somites are going to form? Well, new somites are formed bilaterally, so both left and right at the same time, by periodically creation of epithelial tissue in the presomatic mesoderm. Okay, so at one point, every unit of time, we will see, we will talk a minute about this unit of time, a new fissure is formed in the presomatic mesoderm to define a new somite. Okay, and here in this sketch is quite important. You need to try to understand it. Here we have in the middle the neural plate, if you want, or the neural tube, and here you have a representation of the presomatic mesoderm. You also have a representation of the different somites. The somites are numbered, okay? And usually the somite that is going to be formed in the presomatic mesoderm is called somite 0. So somite 0 is the somite that is going to be formed now from the presomatic mesoderm. Somite 1 is the somite that have been just formed. Somite 2, somite 3, somite 4, so on. But the presomatic mesoderm is also, in the presomatic mesoderm, also some regions are called somite -1, somite -2, somite -3, and -4, and so on. This is because these are the regions of the presomatic mesoderm that are going to become somites after the somite 0 is formed. Then the -1 will become 0. Then when that one is formed 2, the -2 will become 0, and so on. Now, this division, this fissure that is formed in the presomatic mesoderm, establish a boundary, if you want, between the caudal part of the most anterior somite and the caudal part of the next anterior somite and the most anterior region of the presomatic mesoderm. So this is just to say that this fissure, okay, divide or cut off the presomatic mesoderm, forming a new somites here. How do the cells of the presomatic mesoderm know when to divide? Okay, how do they know when to divide and form this fissure? But the model to explain this is called the clock wavefront model. And here is an example, again, from chicken embryo. And as the name suggests, the clock wavefront model has two components, the wavefront component and the clock component. So let's have a look on these two parts of the model. Now, this can be quite difficult. So try to focus. So let's talk about the first part of the model, so the wavefront part of the model. The wavefront part of the model is determined by the opposite gradient of retinoic acid and FGF8. So this gradient form what is called the determination front or wave front part of the model. So if the presomatic cell express high level of FGF, they cannot become somatic. Okay, and then at one point the level of FGF is low enough for the presomatic cell to be able to be competent to become somatic. Okay, so they become competent to respond to the clock. Okay, before that, if the FGF level is too high, then they cannot respond to the clock component of the model, and they cannot become somatic. So the level, the threshold, if you want, level of FGF that allow the cell of the presomatic mesoderm to respond to the clock is the determination front. And this determination front usually is achieved at the -4 somatomer. Now, the most difficult part of the system is actually the clock. Okay? The cell of the presomatic mesoderm experience temporal up and down regulation of notch signaling along the anterior-posterior axis that crash down at the x0 position to form the boundary. So let's have a look on how the clock works using this picture to help us. So here in purple, you have the expression of AD1. AD1 is actually a direct output of notch. So notch controls directive expression of AD1. So the expression profile of the AD1 is exactly the same of the expression profile of notch. Okay, so here we start at the beginning of our clock, if you want time 0. Time 0, we have just formed a new somite S1, and we are defining the boundary of the next somite S0. So if you look at the expression of AD1, which is the same of notch, you see that you have expression at the back of the S1, just ready formed somite. You have expression anteriorly, a bit broadly, but anteriorly in the presomatic mesoderm, and then very broad expression posteriorly in the presomatic mesoderm. Then, while the time is passing, this broad expression posteriorly become more defined and move more anteriorly. While this expression move more anterior in the presomatic mesoderm, the anterior expression of the presomatic mesoderm actually become more defined, and immediately is so defined that it actually define the exact position of the fissure. So at this point, when the presomatic mesoderm expression notch is moving anteriorly, the fissure start to form. Then after a while, when this fissure is finished and a new somite is formed, so you see a new S1 is formed, the posterior expression of notch in the presomatic mesoderm is completely disappear and just move in the anterior part of the presomatic mesoderm. So this basically is the last point of the clock, if you want, point 24, and then we start again with time 0. Okay, so the period of this expression, of this clock, if you want, is in the chicken embryo 90 minutes. And you can see that from time 0 to time 90, if you want, from time 0 in your clock to 24 or 12, as you want to call it, you go and find yourself in the same expression profile that you had at the beginning, where you have expression more posteriorly and in the presomatic mesoderm, broadly, and you have expression anteriorly, but not quite defined enough to be able to induce the formation of the epithelial fissure. Okay, now take 10 minutes break, 15 minutes break. You deserve it. That was a difficult part of the lesson. Perhaps have a hot beverage, and if you want, take the quiz that I put on Canvas, quiz number 1.